Hi, my name is Raylan Grabill. I'm a junior biology major here at Cal U, and during my undergraduate research here, um, I really developed a passion or an interest in studying the antibiotic crisis and how bacteria are able to become resistant to our antibiotics that seemed to prove themselves as the most effective way to treat infections or disease. And overall, there's definitely a noticeable decrease in effectiveness of our traditional antibiotics, and it's becoming a global public health problem. So, understanding how bacterial cells communicate and behave can definitely pave way for new therapies to combat these deadly antibiotic resistant pathogens that we're experiencing in our public health. Um, so, bacteria communicate using quorum sensing, which involves chemical signaling molecules that assist in synchronizing cellular activity and behavioral responses. And in bacteria, chemical communication involves producing, releasing, detecting, and then responding to small hormone-like molecules called autoinducers. And quorum sensing in bacteria allows for biofilm formation and coordinated virulence against host infections. So, by gaining an understanding as to how quorum sensing controls this bacterial behavior, we can further develop strategies to disrupt the cellular communication and decrease that biofilm formation or decrease that bacterial growth or combat this virulence, leading to new mechanisms to kind of combat these antibiotic resistant pathogens without trying to find new novel antibiotics to treat them with. So, to understand how they're able to communicate to form biofilms or how they coordinate their virulence in their host, you have to understand the process of how they communicate in general. So, for bacteria to communicate, they must possess three characteristics. They have to secrete a signaling molecule, which is that autoinducer, and they have to be able to detect the change in this concentration of the signaling molecule and then regulate gene transcription as a response. So basically, one, two, three bacteria on their own cannot create this quorum sensing communication. So what happens is when there is a population density, a high population density, then more of these autoinducers are going to be able to be secreted by the bacteria. That means more are going to be able to be detected, and a response is going to be allowed to occur to regulate gene transcription. So with a low cell density, this quorum sensing circuit can't take place. But with that higher concentration or higher cell density, the quorum sensing circuit can take place. And so through this signaling of molecules, detecting it and inducing a response by regulating transcription, they're able to activate either biofilm formation or coordinate their virulence. So I did two examples here. The first example under biofilm formation showcases Pseudomonas aeruginosa. And this diagram basically shows that Pseudomonas aeruginosa is a pathogenic bacteria that causes pneumonia and takes more effect on patients with cystic fibrosis. And so basically with biofilm formation, cells have to adhere and attach, and then they have to colonize and form this biofilm. And in this biofilm, they're able to function together as a group, and they really can't be harmed as they're encased in this extracellular matrix that they form by joining together and communicating. And our antibiotics that we have created it's really hard for those antibiotics to penetrate this biofilm formation, therefore making it hard to treat these infections caused by these pathogens. So bacteria also communicate to coordinate their virulence. And down below in figure 3, this shows an accessory gene regulatory system in Staphylococcus aureus. And I'm not going to walk through all the steps in this process, but basically in a lump sum, again, this is when there is a high concentration of bacteria secreting the signaling molecule, in this case an AIP. And whenever that's secreted, then a response can take place. And this response is the secretion of virulence factors. And the example here shows Staphylococcus aureus, which is an infectious pathogenic bacteria that causes MRSA, which we see becoming antibiotic resistance in healthcare. And so we can look at these cellular communications and their responses, whether that be biofilm formation or coordinated virulence, and we can ask ourselves questions. Where do we go next? Like, what's the next thing? Well, with biofilm formation, we want to ask ourselves, how can we inhibit biofilm formation from even taking place? And one area that I focused on is using quorum sensing inhibitors. But what are those quorum sensing inhibitors, and how, like across the board, do all quorum sensing inhibitors work in every case, or is there different inhibitors for specific pathogens? So like a lot of questions of these arise, and that's definitely where I'd like to delve in more in my future research. But also with Staphylococcus aureus, we looked at the AGR system, or the accessory gene regulatory system, and how they secrete these virulence factors. But how do we eliminate that secretion? How do we stop that secretion? Because in turn, if we stop that secretion, we stop antibiotic resistant infections from these pathogens. But overall, I want to look at a bigger picture of what approaches can we take to counteract the overall crisis of antibiotic resistance. And from this understanding of how cells communicate and how they behave and cause human disease, there's definitely been more prospective therapeutic techniques that can be explored and implemented. For instance, with quorum sensing inhibitors, it has already been provided evidence as an alternative method for fighting bacterial infections. However, finding quorum sensing regulatory systems that are critical to this microbial pathogenicity and drug resistance should definitely be a major focus in the future research of combating these antibiotic resistant pathogens.